Assalamu alaikum, welcome guys, you are watching Pre and Course channel. As you are acquainted of the fact, I have been delivering online lectures for class 12th and NEET, but today I am going to make an outer churning video. The video will serve the purpose of those students who are pursuing higher studies. So what actually I am going to deliver today is the same basic topic, that is sex discrimination in case of human beings. We already discussed it in case of our previous video, but today I am going to discuss it at embryonic and gene level. Apparently or superficially, it's very simple. We all know it that as the Y chromosome over there which is determining sex. But what actually happens in the background of it, that is to be seen. What the mystery, how gene operates there, what is going on, how is the development taking place, that is to be seen. So let's break that mystery. Stay tuned, watch this video till end. You will definitely come to know some new points today, inshallah. So let's start. So let me tell you, when there is a fertilization, zygote is formed. Then this zygote undergoes the cleavage, then different processes. There is morulation, blastulation and gastrulation is there. And after gastrulation we get three types of germ layers. We got the endoderm, mesoderm and ectoderm, right? From these three germ layers, primitive germ layers, we get all other types of organs over there. So my concern here is yolk sac is one of my concern, and these are some organs, precursors. These are my other concern. So as you know, the embryo is developing. At the same time, we are having the extra embryonic membrane that's also developing. We got the chorion, amnion, allantois, yolk sac, they are also developing at the same time. So we got this yolk sac over there. This yolk sac is developed, it has got the origin of endoderm and mesoderm. It is originated from the endoderm mostly and partly from the mesoderm. On the other side, we got this intermediate mesoderm, it gives rise to different organs. Some of the organs are these are the precursors of different male reproductive organs or female reproductive organs. So let us see what happened at the fourth week. At the fourth week of fetal life, in case of yolk cells, primordial germ cells are formed. These primordial germ cells are the precursors of the gametes, sperm or ova. Okay, depends upon its genotype, X or XY. On the other side, at the very same time, that is the fourth week of fetal life, we got some different organs. We got the gonadal rudiment is there, Wolffian duct is, Mullerian duct, genital tubercle. Let's see, the gonadal rudiment is there. This gonadal rudiment, it's actually the precursor of the gonads. It's the precursor of the gonads. As you know, if for example an organ, a precursor has to make a lung, so we need to have lung rudiment. We have lung rudiment in the body that will form the lungs. Similarly, we have got the heart rudiment that will form the heart. And in this case, we got this rudiment, gonadal rudiment over there. Gonadal rudiment will form what? It will actually form the gonads. So what is this gonadal ridge? So we don't have to be confused. Let us be clear over here. This gonadal rudiment, its upper ventral part is actually made up of the epithelial tissue. It is the epithelium over there, genital epithelium, gonadal epithelium, and we call it as the gonadal ridge. It is actually the precursor of the gonads. So let's see what happens. So I told you at the fourth week these germ cells are formed over there, right? Germ cells are formed. So they will start migration. They will migrate. The germ cells will migrate from yolk sac. They will migrate towards the gonadal ridge. Let's see what happens. So primordial germ cells, they will migrate from yolk sac towards gonadal ridge. Okay. Until the 7th week of fetal life or 6th week of fetal life, this gonadal ridge is undifferentiated. It's totally undifferentiated. You cannot differentiate whether it's going to be a male or female at that time. So what happens next? So what actually happens was there, this gonadal ridge is actually, I told you, it is the upper ventral part of the gonadal rudiment. So this is the it's made of the epithelial tissue. This is the epithelium. It proliferates deep into the loose connected cells, that's called mesenchymal cells, which are present in the gonadal rudiment. So don't be confused. Mesenchymal cells are actually the cells which are of mesodermic origin. They are having the multipotent to develop into different types of cells. So this gonadal ridge, which is the ventral portion of the gonadal rudiment, it will proliferate, it will grow deep down into the loose connective tissue, mesenchymal tissue of this gonadal rudiment over there. Okay, got it? So this will actually form, then they will fuse together these epithelium tissue inside the mesenchymal tissue, inside the loose connective mesenchymal tissue of the gonadal rudiment. It will actually fuse together and form the sex cords. It will form the sex cords. Let's hold on the migration process for some time. It is gonadal ridge. What will it do? We also call it as a bipotential gonads, because it has got the capability to get converted either into the male gonads or female gonads, testes or ovaries. So what will happen? They will form sex cords. When this gonadal ridge proliferates deep into the mesenchymal tissue of the gonadal rudiment, these epithelial tissue will fold together and they will form the sex cords over there. These sex cords, they can be of testes cords or cortical cords, right, depending upon the invasion type of germ cells which are invading it. If XY type of this germ cells are over there, it will form the testes cords. If we have XX type of germ cells, we get cortical cords over there. So what actually happens? For example, let's see XY type of germ cells, they will invade this gonadal ridge. This gonadal ridge, in case of XY type, it will proliferate deep into the loose connective tissue in the interior of it, in the interior of the gonadal rudiment. That's called a medulla. That's we call as medulla, and there it will fuse together. They will fuse together and they will form these sex cords that we call as testes cords. And these are the testes cords which actually form the type of cell. They are known as Sertoli cells. Okay. So adjacent to this, we got some mesenchymal cells also there, because it's growing deep into the mesenchymal cells. So in the surroundings there are the mesenchymal cells. So mesenchymal cells will be there. This we could say over here, interstitial mesenchymal cells. They will develop into Leydig cells. Okay, they will develop into the Leydig cells. So let us see. So testes cords form the Sertoli cells. Let us write it over there on the top so that we can we have got these testes cords. They will form the Sertoli cells, and on adjacent to this we have these mesenchymal cells. I told this mesenchyme is a simply tissue of mesodermic origin, has the multipotential to develop different types of cells. They will form the Leydig cells. Okay. So these Sertoli cells will do what? They will secrete AMH, anti-Mullerian hormone. they will secrete AMH, anti-Mullerian hormone, and this will cause regression of Mullerian duct. This is called the regression of the Mullerian duct. You know, the Mullerian duct is actually the precursor of the female accessory reproductive organs. It actually forms, in case of female, it forms uterus, it forms oviduct, it forms upper part of the vagina, it forms the cervix. But here, in case of males, because of the secretion of this AMH, anti-Mullerian hormone, this got regressed, degeneration. It is degenerated. Okay, so this is degenerated. While as these Leydig cells do what? They will secrete testosterone. When they will secrete testosterone, this testosterone will cause differentiation of Wolffian duct. Into what? Into vas deferens, epididymis, spinal vesicle. So these are actually the male accessory reproductive organs over there, ducts over there. Vasa deferens, epididymis, spinal vesicle, they are all differentiated from the Wolffian duct under the impression of this testosterone hormone. So vasa efferens is missing over there. Vasa efferens is actually the remnant of the mesonephric kidney, which is already present there. In case of females, when XX type of primordial germ cells, they will invade gonadal ridge. What will happen in that case? What will happen? Let us see. In that case, this gonadal ridge, as I told you, is proliferates into the mesenchymal tissue of the gonadal rudiment, but here it will not go deep. It will remain at its surface, but it will proliferate deep into the mesenchymal tissue, but not into the deeper level, but at the superficial level. So it will be surrounded by the mesenchymal tissue over there, but not at the deeper level. It's at the cortex level. So that's why we call it as a cortical cords. That's why the sex cords, here we call it as cortical cords. So this cortical cord will do, what will it do? These cortical cords will secrete estrogen. These cortical cords will actually differentiate the type of cell. They are known as granulosa cells. And furthermore, these cortical cords are surrounding the what we call the future egg over there, ova as well. So these are differentiated into the granulosa cells and the mesenchymal tissue, adjacent to mesenchymal tissue, which differentiated into Leydig cells. In case of males, here it will differentiate into theca cells. Okay, both these cells will join together and they will form follicles. Okay, and these follicles will be surrounding an ova. Cluster of follicles will be formed in this manner, and this cluster will be containing one ova, and these follicles will finally contain the ova. It will secrete estrogen. They will secrete estrogen over there, and this estrogen hormone, as there is no testosterone hormone which could cause, there is no AMH hormone which could cause the regression of the AMH. So AMH will be Mullerian duct. This Mullerian duct will be differentiated. And the Mullerian duct will be differentiated into into what? Into oviducts, into uterus, similarly into cervix, and into upper vagina. This malurin duct will be differentiated because there is no AMH to cause its regression, no AMH over there, so it will be differentiated, and because of this estrogen on its impression as well, so this will be differentiated. And second thing, what will happen? This estrogen will also cause the differentiation of genital tubercle into labia, vagina, lower part, and clitoris. So one thing I forgot to tell you, in case of males, what happens when under the impression of testosterone, Wolffian duct is, this I told you, Wolffian duct is differentiated. Another thing, this testosterone gets converted into DHT, dihydroxytestosterone. In case of in genital tubercles, it gets converted into DHT in किस में? In genital tubercle. Okay. Through an enzyme, the enzyme is known as 5-alpha reductase, or actual name is 5-ketosteroid reductase 2. Okay. So under this DHT, dihydroxytestosterone, this will cause the differentiation of this genital tubercle into penis, prostate, and scrotum. Okay, this is what happens actually at the embryonic level. But now let's see what happens over there at the gene level. What happens at the gene level? That's what we see. So here let's see what genetically happens over there. In case of XX and XY, in case of XX chromosomes, these chromosomes, we got the DAX1 genes, DX1 gene, one DX1 gene add on each copy. However, in case of X gene, we have got DX1 gene and SRY gene on Y chromosome, and this SRY gene is dominant over this DAX1 gene. It suppresses the activity of DAX1 gene. However, when there are two DAX1 genes or there are two copies, they will show their expression and female will be formed. So let us see, this is not the all story, this is one side of the story. Let's see what there are some autosomes as well which are supplementing the role of this sex determination. In case of males, SRY gene is there. This SRY gene is present on chromosome number, that's X. SRY gene, sex determining region, Y gene. SRY gene is present on the short of all the Y chromosome. This gene will activate SF1 gene, and this SF1 gene, in presence of this SRY gene, these two genes will activate, upregulate, and express SOX9 gene. Okay, SOX9 gene, in presence of some other genes like SF1 plus WT1, Wilson tumor gene, what they will do, they will actually upregulate the expression of AMH gene. This AMH gene will produce the protein that is anti-myosin Mullerian hormone. This AMH will cause a regression of Mullerian duct. Nice. Okay. On the other side, this SOX gene will activate F9 gene, and this will cause the secretion of testosterone. And testosterone will lead to the differentiation of Wolffian duct and indirectly of genital tubercle. And this FGF9 gene will also antagonize the activity of WNT4 gene, okay, which is actually the ovary determining gene, autosomal gene present in both sexes. So this is what happens in case of males. So what happens in females? In females, we have DAX1 gene. This will activate, DAX1 gene will activate WNT4 gene and RSOP1 gene. These two genes together, they will form a product that is called beta catenin, and this beta catenin will inhibit, will suppress the activity of SOX9. suppress करेंगे इसको, they will activate, suppress or inhibit the activity of SOX9. So when there is no SOX9, there will be no AMH, so no regression of Mullerian duct. So Mullerian duct will be formed. On the other hand, this WNT4 gene will also cause the suppress the activity of phospho alpha reductase enzyme, which is very important for conversion of testosterone into DHT, dihydroxytestosterone, which finally causes the differentiation of genital tubercle into penis, male genitalis, penis, prostate, and this scrotum. So this will actually inhibit the activity of phi alpha reductase enzyme as well. Okay, and the other side, this WN4 will also help in the determining of the production of the estrogen hormone. That will cause the differentiation of the Mullerian duct or genital this tubercle over there. This is all about the genetic gene level differentiation, what happens at the gene level. So now we will discuss 2-3 cases which were reported. One of the case was reported in 1980. In that case, there were two sisters, twin sisters. They came, and it was reported that both of them were containing the XY type of genotype. XY type genotype. However, we know XY genotype actually determines male sex. Both the sisters were containing XY type genotype, but both were females. There was a female phenotype over there. Genetically, genotypically, they were actually the XY type. So what had happened over there? So when it was analyzed, when their sex cells were studied, they came to know what is over there. What has happened over there is that they had this XY type. These are 2 chromosomes. Y is a bit shorter, and usually we have DAX1 gene on Y and we have SRY gene on, DAX1 gene on X and SRY gene on Y chromosome. But in their case, there were 2 copies of DAX gene. 2 copies of DAX gene were present on one X chromosome, and this caused the suppression of the activity of the SRY gene present on the Y chromosome. So that's why their phenotype was of female phenotype, although they were having the male genotype. This is one case. In second case, which was reported in 1991, a person having this XY genotype, again, but the baby having the XY genotype have got the blind pouch vagina. Okay, there are no penis, there are no what we call the male genitals over there. However, the internal structure, the Wolfram duct had differentiated into the vas efferents. What difference was this seminal vesical epididymis. What had happened over there? Actually, the enzyme, as I told you, this testosterone is converted into dihydroxytestosterone through an enzyme. The enzyme is 5-alpha reductase, or the full name is 5-ketosteroid 2. So this enzyme was missing. Due to the absence of this very enzyme, the testosterone was not converted into DHT. Because of no DHT over there, genital tubercle had not differentiated into the male genitals. Okay, in such patients, in such babies, they are raised as females because of their absence of the male genitals. But when they reach the age of puberty, due to the over secretion or the good amount of secretion of this testosterone hormone, these receptors will respond to the good amount of the testosterone hormone, and they will finally differentiate into the male genitals. So this very baby will definitely at puberty, it will show its male genitals. It will excel its male genitals. Male genitals will differentiate. So this was the second case, because of which we came to know that this 5-alpha reductase enzyme is very important for conversion of this testosterone into the dihydroxytestosterone, and this dihydroxytestosterone which is actually responsible for the conversion of genital tubercle into male genitals. This is very important. This is very important over there. Right. The third case was reported, in which what was done, not reported actually, it was an experiment done. Experiment was done in case of the newly formed zygotes of mice having the genotype XX. The SRO gene, part of the DNA containing the SRO gene, this part of the DNA was micro-injected into the zygotes of those mice containing the XX type genotype. When they were born males. They become male mice because of this SRO gene. However, they were having the genotype as XX, but because of the micro-injection of this SRO gene, they turn into the males. Okay, hope you have got this. This was all about the sex discrimination in case of the human beings at embryonic and gene level. There are so many books over there. There are so much literature is there. New things are emerging every day. So you can adapt to this knowledge. And if you know something more than this, you can comment so that I can also learn. Thank you very much.